Alright, so this is a collaborative study that's got members from University of Adelaide, CSIRO and SARDI. It's being supported by JUNA and obviously funded by Goiter. The project's ongoing and it's scheduled for completion in June 2015, so we actually don't have all our results back in yet because there's a lot of laboratory analyses. So I'll largely focus on the background methods and sample collection for the study, but I'll also present some preliminary results as well. So estuaries represent really unique environments. They're the mixing zone between freshwater and marine systems. And as such, they're highly influenced by freshwater flows. So freshwater flow can dictate the salinity regime of estuaries as well as the water level within them, and it also influences connectivity in relation to hydraulic and biological connectivity. Now when we talk about connectivity, we often focus on the movement of biota and particularly fish, but connectivity is also really important in regards to the transfer of energy and nutrients between these systems. Now hopefully that's clear enough, but this conceptual diagram really just explains that process. Effectively, riverine discharge carries with it organic matter and biota such as phytoplankton and zooplankton into estuaries. Now that organic matter, or even the biota that are transported to the estuary, or in the case of the question we just received, into the ocean, may be consumed by estuarine biota, and as such, carbon that's actually generated in a freshwater or terrestrial ecosystem can be assimilated into an estuarine food web and thus subsidise the food web and enhance secondary productivity. However, a problem facing a lot of the world's large rivers is that river regulation and consumptive water use results in reductions in the amount of fresh water that are flowing to these systems. So the Coorong is one such system that may be suffering from these impacts. It sits at the end of the Murray-Darling Basin, which has large upstream storages and large levels of irrigation throughout the basin. And as such, there's been severe reductions in freshwater discharge to the Coorong. And now, on average, around about a third of natural flow reaches the Coorong on an annual basis. Furthermore, a series of the 5 tidal barrages were constructed on the flow points between Lake Alex and the Coorong back in the 1930s. And these now act to control discharge between these two systems. Now we've got quite a good idea about what reductions in flow has done to the salinity and water level regimes within the system, but I've got no idea what impact that had on productivity. And perhaps more importantly, we've got very little idea on how freshwater inflow influences trophic subsidy within the Coorong in a contemporary sense. Like Chief Eng highlighted before in her talk, the Coorong and the basin more broadly has undergone some really variable hydrology in the last decade or so, and that included a prolonged drought period, and then that was followed by a period of a few years of quite high flow. Now fortunately through this period there were several projects undertaking monitoring in this region, so we got a real good handle on what biological patterns were occurring in the region. And indeed they were quite stark between the different hydrological periods. In regards to fish, there were quite stark changes in the presence and abundance of a range of different species, and also similar patterns were seen in zooplankton and phytoplankton between the drought and high flow periods. And indeed for both of these groups of animals there was generally increased diversity, abundance and productivity during the high flow periods. So when we look at some of these patterns we start to get an idea about processes that might be underlying these patterns, and we can start to make up some hypotheses. So one such pattern that we noticed was that of this small species of fish called the sandy sprat, but you may know it from the bait shop or the Greek restaurant as whitebait. And this plot here presents the abundance of this species from 2006 to now. Long story short, the species is much more abundant during times of high flow to the Coorong. And to really hammer that point home, the y-axis on this plot is actually split, so the abundance during those high flow years is orders of magnitude greater than in low flow years. So then we go back to the life history of this fish and what do we know about it? Funnily enough, it's actually considered a marine species. So in southern Australia it's found in sheltered habitats within the Gulfs, but it's also found within the Coorong, and indeed it's highly abundant in the Murray Estuary region of the Coorong. It's probably the most abundant small-bodied fish in the region. It's a member of the Clypeid family. It's planktivorous, feeding primarily on zooplankton, and in turn, pretty much everything else eats it. Whilst there hasn't been studies in the Coorong about the diet of these birds, it's been shown to be a really important prey item for species along the southern coastline, including terns and little penguins. But recently in the Coorong, a study that Chief Eng and others at SARDI have recently been doing, it's been found to be consumed by many piscivorous species of fish in the Coorong. And indeed, for mulloway, less than 400 mils in length, it's the most important prey item for this species. So overlaid on top of this was the data I spoke about before from during the flood, and that high flows, we know, have a large influence on the zooplankton and phytoplankton communities in relation to diversity and abundance. These things eat these guys, so it leads us to hypothesise that is maybe freshwater-derived carbon influencing prey availability and abundance of sandy sprat, and thus subsidising the Coorong food web. Now, we sit at a pretty pivotal point in time in the management of the Coorong and Lower Lakes, and more broadly the basin. The proposal and implementation of the basin plan was met with nothing short of contention in a lot of communities. So I think perhaps more importantly than ever is that we have empirical data on the influence of flow on ecosystem function with which to support management and future delivery of environmental flows. And really that's the ultimate aim of this project. When this project sort of kicked off, we had to look at what the flow forecast was for this year, and unfortunately it was looking like a rather below average year. But nonetheless, in dry periods, these are the kind of volumes of water that we might indeed have to argue over, so they're just as important as higher flow volumes. So in particular, we wanted to investigate productivity and trophic subsidy in relation to these low volume flows to the Coorong. And specifically, we wanted to investigate the influence on water quality and primary productivity in relation to phytoplankton, microbiota community structure and particularly zooplankton, also the abundance and diet of sandy sprat, and last but not least, the assimilation of riverine organic matter through the food web up to sandy sprat. So we focused our research in the Murray Estuary region of the Coorong. Given the low flow volumes, this was really the part of the Coorong that was likely to be most influenced by low flow volumes. And we sampled a range of sites through this area in spring, summer just gone. At each of these sites, water, fish and zooplankton were collected, and they were brought back to the lab to undergo a range of traditional analyses. So things like looking at algal counts, zooplankton counts, and looking at fish gut contents in the laboratory. To complement these techniques, the DNA sequencing is also being undertaken to look at species that are present in water samples, zooplankton samples, and again in fish guts. Now, the last aim I spoke about before was about the assimilation of organic matter of riverine origin into fish tissue, and we're doing that by the use of stable isotope analyses, and I've actually got some of the results for that, and it's quite a complex process, so I'll run through that a bit further. Basically, all animals acquire an isotopic signature of nitrogen and carbon, and they acquire this signature from their food sources. They don't require it from anywhere else. And unlike a gut content study where you're looking at the diet of a fish from potentially the last few hours or luckily, if you are, days, the signature that a fish acquires with stable isotopes integrates diet over quite a long period. And it's probably best or easiest to explain this process by looking at that graph on the right, which is a typical nitrogen versus carbon plot that you get with these analyses. That is real data. That's data that was collected during the clam ecology project in 2007 from the Coorong, and each of those dots represents a different species. Now, in particular, with carbon, predators acquire the same signature as their prey. So if something is eating something else, it effectively should sit above it on one of these graphs. So these 2 points I've highlighted are sandy sprat and mulloway. And again, this corroborates our other data that these fish do prey upon the other. Perhaps what is more important to the current study, however, is that you can determine the origin of organic matter that's fueling a fuel web from these analyses as well. And how you're able to do this is that carbon which is generated in a freshwater or terrestrial environment is typically depleted in carbon, in contrast to marine sources which are typically enriched in carbon. So this plot is probably not the best example given this data was collected during the drought when there was no flow, but if you can imagine, if you have an animal which is deriving its energy more from a freshwater source, it should sit more to the left of this graph, and marine, vice versa, more to the right of the graph. Now, nitrogen actually performs quite a different function. Effectively, nitrogen is enriched as you move up a food chain. So the way nitrogen works in these analyses is that it can allow you to determine how many links you have within a food chain or exactly where a species sits within a food chain. So as I said, a range of analyses are still underway, and they include some of the traditional laboratory stuff we're doing, so algal and zooplankton community composition and abundance counts, the sprat diet stuff and the DNA sequencing. All of this stuff is still underway. However, we recently got the stable isotope analyses back for the isotopic signatures of our fish muscle tissue, and it's hot off the press, so I'll quickly go through that stuff now. But first of all, just to put it in a hydrological context, this is the flow or discharge from the barrages that we experienced this year. Since September, it's been, give or take 1,000 megs a day, around about 2,000 megs a day have been flowing out of the barrages and going up and down a little bit, but it's reasonably consistent. Nonetheless, there was a pretty significant flow peak in August, which peaked at about 20,000 megs a day, and we sampled in November, late November, and then again in December. And the importance of that flow peak will be apparent a bit later. So I've simplified the data here because this makes it a bit easier to explain. This is again one of the typical nitrogen versus carbon plots that we get from these analyses. Nitrogen on the y-axis, carbon on the x-axis, and it's really the carbon data that I'm most interested in presenting to you today. I've actually averaged the values for each of the fish that we sampled in each of our trips to give us 1 point for each trip. And interestingly, as you can see in sort of chronological order, there seems to be a gradual enrichment in carbon in the signatures in these fish as you move from left to right. And these error bars on here are actually standard deviations, not standard errors. So it appears that, as I was saying before, with freshwater carbon sources and marine carbon sources are quite different, and freshwater are more depleted than marine sources. So it does appear that potentially, as time has gone on, that these fish are showing less of an influence of freshwater carbon in their diet. And what this might be showing is greater time from the flow spike that we saw in August. Nonetheless, interpreting these data on their own is a bit difficult, and it's more interesting to look at this data in reference to other data that we've got. And fortunately, we do have some stable isotope data from the same species from different time periods. So these next two dots, the blue one actually comes from March 2004 from a recent honours project. And it was collected, funnily enough, in a period that was very, very similar hydrologically to the current study. The other data point we have over to the right was collected during clam ecology during 2007 during a period of no or very, very little discharge to the Coorong. And as you can see, the carbon isotopic signature of fish from those samples sits much more enriched in carbon towards the right, suggesting that those fish were deriving much of their energy from marine source, and that during low flows that we experienced this year and during March 2014 was indeed enough to show that freshwater flows do seem to be subsidising the trophic web in the Coorong to some degree. Nonetheless, these results are very preliminary. We've got a range of analyses that are still to come back in, and hopefully that'll clarify the story a little more. So as I said, there's a lot more still to go, but when we get all that stuff back, and it's particularly analysed in regards to other data that we have on similar parameters from different hydrological periods, it'll hopefully facilitate a much greater understanding of the influence of hydrology on a range of parameters in the Coorong, including phytoplankton and zooplankton. Perhaps most importantly, it'll give us much more data on the influence of freshwater and terrestrial carbon on the estuarine food web. And also, these have been knowledge gaps that have been brought up in a range of different projects, including a previous Goiter one. So these will answer some of these knowledge gaps. And really, I guess the ultimate goal of the project was to provide some of this empirical data that I spoke about before that I really think is quite important for supporting management and cases for going to water holders and arguing for water. So this should provide some of that empirical data that support these future environmental water bids, and given that it was done at low flows, this will be particularly applicable to times of water scarcity. It also acts as some of the first kind of research of this type that we've done within the Coorong. So it can really be built upon much further, and if it's done at a range of different flows, we might be able to quantify outcomes for different volumes of water much better. And in the end, if it all works out well, hopefully this research can influence basin plan watering strategies and priorities into the future as well. Thank you very much.